All right, are there any questions? So last time, this is where we start, okay? Yeah, go ahead. Uh-huh. Of course, yeah. Otherwise, you'll get skin cancer, you know. Yeah, we have repair mechanisms to repair that. It'll go and cut out this portion where the dimer is there, okay? It'll cut out this portion, look at the complementary strand, make a copy, right? Fix this portion, and then it's repaired, right? We do have enzymes to repair that, repair these thymine dimers. Otherwise, you will get, sunburn and skin cancer. Okay. Now, before that, there is one more item of business that I need to do, right? Because the federal government wants to make sure that you guys are attending the class. Okay, you didn't sign up. Okay, I don't know, maybe for keeping track of scholarships or whatever. So let me just take some attendance. Okay, Jordi Abante Linas, is you there? Okay, good. Ahmed Al Kawam, there. Shaheen Baluki, Chun Chi Chen, Shabnam Dagagi, okay. Yusuf Dogan, Mahdi Imani, Hondo Jiom, there. Hyun Chol Jo, Ali Reza Karbala Khare, okay. Yunji Kim, Suman Priti, and Sajat Zadeh, or Sadeh, Huang Min Tran, Zhao Wang, Shulian Si, okay, and then Siamak Zamani Dadan, okay. So everybody is here. Yeah, right now the requirement is on us. Okay, we have to go on Howdy and just certify that everybody is attending. So I don't know what they are going to do. But maybe not for you guys, but for undergrad students, if they're getting scholarships and things like that, they want to make sure that you are actually taking the class. Not attendance for this class. Like they mentioned something like one way would be to collect homeworks or exams and keep track of whether people are really doing the class or not. Huh? Not yet. Yeah, that's the reason I had to do the roll call. Yeah. Okay. So yeah, getting back to this genomics thing, like every time you step out into the sun, all right, if you have two adjacent thymine bases like this, all right, on one of the DNA strands, you will get a dimer that's going to be formed, all right. There is some kind of a linkage, right, covalent linkage between these two thymines. That's not good, right. So we have repair mechanisms, all right, that'll go ahead and cut out that thymine dimer, all right, and then look at the complementary strand and make a copy, right, and repair this. Now, nearly all of the repair mechanisms depend on the existence of two copies of the genetic information, one in each strand of the DNA double helix. Most DNA damage creates structures that are never encountered in an undamaged DNA strand, and this is thought to facilitate the identification of the damaged DNA strand. Which one is the strand that is not damaged and can be used as a template for repair purposes, and which one the damaged DNA strand is. You need to be able to distinguish between these two, and that probably is thought to be facilitated by these special structures that DNA damage creates. So the basic pathway for repairing damage to DNA involves the following three steps. In the first step, the DNA damage is recognized and removed by one of a variety of different nucleases. All right. Again, nucleases are enzymes that degrade nucleic acids. They will go, if you give it a nucleic acid, you treat a nucleic acid with a nuclease, it will go and cut up the individual nucleotides. Right. Then after that, a repaired DNA polymerase will bind to the 3' hydroxyl end of the cut DNA strand, right? And it will fill in the gap by making a complementary copy of the information that is stored in the undamaged strand, which is going to be used as the template, right? So when the repair DNA polymerase has done its job, right, there will still be a break in the sugar phosphate backbone of the repaired strand. And this nick in the helix is sealed by DNA ligase, right? So ligate means to stitch, so DNA ligase is going to stitch the two strands together, and then you have the repaired DNA strand. Now, failure to repair damaged DNA can result in very serious diseases. For example, we all have the enzyme, right, to go and repair those thymine dimers. Okay, like getting back to your question, every time because you go out into the sun, right? I mean, you don't just stay indoors, right? And you're still fine. So that means you must be having the enzyme to repair that damaged DNA. If you don't have that enzyme, right, then you can get a disease that is called xeroderma pigmentosum. And people who have that disease, they cannot get out into the sunlight, right? Kids who are born with that disease, they have to play by electric lighting at night, right? They cannot go out and play in the sun, okay? And it's a pretty serious disease, and ultimately they can get skin cancer. So they are prone to severe sunburn and skin cancer. Yeah, go ahead.
The DNA repair the molecule by the complement part. So if the pair
damaged, then you're in trouble. Okay. Yeah, if you have if somebody subjects you to a heavy dose of radiation, right, where both your strands get messed up, then you're in trouble. You know, then you might get cancer pretty quickly. Okay, because you cannot repair. Now, to conclude this chapter, we note that the high fidelity with which the integrity of the DNA is maintained means that closely related species are going to have very similar DNA sequences. For example, humans and chimpanzees have DNA sequences which are about 98% identical. You go and compare, you'll see, I mean, there's a very big degree of matching. Even apparently unrelated species such as humans and whales, they share a lot of similarity in their DNA sequences, probably to the tune of like 90% or something like that, right? And this strengthens the hypothesis that all these organisms must have originated from a common ancestor at some point in evolutionary time. Okay, nobody knows for sure, but the DNA sequences are so similar. All right, any questions about DNA? This chapter on DNA is now over, all right? So DNA actually codes the information that is needed for making proteins. So in this chapter, all right, we will see how the information from DNA, right, is used to make the proteins that the cell needs, right? So we are going to cover what is called transcription and translation, right, in this chapter. Now the DNA contained in the genome of an organism primarily codes instructions for making proteins. Recall that there are only 4 DNA bases, right? Adenine, guanine, cytosine, and thymine, right? So an instruction that is written out in the language of DNA is basically an instruction that is written out in a language that has a 4-letter alphabet. So if you look at the human genome, you have 3 billion of those letters, okay, and they're spelling out some biological message, okay. That has all the information that is needed to produce you and me, okay. Those 3 billion letters, okay, your eyes, nose, ears, everything, right, can be produced from that information, right. So the code is there. On the other hand, you know, so the DNA, the information in the DNA actually codes for proteins, right? So proteins are made up of amino acids that are linked together by peptide bonds, and we have already studied these, right? Since there are 20 possible amino acids that occur in nature, right, the amino acid sequence for a protein can be thought of as a biological message that is written out using letters from a 20-letter alphabet, right? There are 20 amino acids, there are 4 different DNA bases, okay? So you have to figure out the mapping. For those of you that do coding theory and all that, you know, you probably know that, you know, if you have this many digits, you can do that much of coding, all right? So nature has already figured all that out, you know. So natural question comes up, how does the DNA message coded in the 4-letter alphabet map into the amino acid sequence of the appropriate protein? So in this chapter, we are going to provide a detailed answer to this question, right? How you go from DNA to protein, right? And it goes through 2 steps, all right? In the first step, you get RNA, and then you get protein. The DNA does not direct protein synthesis by itself, but it acts through certain intermediaries, right? So directly you don't go from DNA to protein. When a particular protein is needed by the cell, the nucleotide sequence of the appropriate portion of the DNA molecule is first copied into another type of nucleic acid, which is called RNA, which we already encountered. It is ribonucleic acid. It's similar to DNA, except that the sugar is different and the bases are a little bit different, right? Now, RNA copies of short segments of the DNA are used as templates to direct the protein synthesis. So it's like the DNA is like the master copy, okay, that should not be changed, okay, it should remain unmutated, unaltered. Every time a protein is needed, you take the master copy, run an intermediate copy, get that information, and then from that copy you produce the protein, right? So the process of copying the DNA into the appropriate RNA strands is referred to as transcription, right? We are going from one language to another. And the process of producing the protein from the information in the RNA is referred to as translation. Now some terminology. Again, we said a gene is roughly a stretch of DNA that codes for a protein. When a gene is being transcribed, that means that the DNA in the gene, right, is getting copied into RNA, right? Then we say that the gene is being expressed or that the gene has been turned on. Okay. This is just like computer code, okay? Like you might have a lot of code, a lot of it is not active at all times, okay? So you selectively turn it on and off. Now the usual flow of genetic information in all living organisms is from DNA to RNA to protein, right? And this fundamental principle is referred to as the central dogma of molecular biology, right? Because all living organisms, they express their genetic information this way, from genes to RNA to protein, right? There are exceptions, like if you look at the AIDS virus, that is a retrovirus, right? So it has RNA, it will transcribe that into DNA and then it will invade your system, okay? So there are some exceptions, okay? But, you know, for all practical purposes, most living organisms, the information flows from DNA to RNA and then to protein. So this is called the central dogma of molecular biology. All cells on this planet, right, from the simplest bacterium to complex organisms such as humans, will express their genetic information in this way. Yeah.
Why not just from DNA to protein?
Why not? I don't know. Okay.
They are little different.
Well, I mean, because see, if you have one master copy, one of the reasons could be if you have one master copy, you're going to run copies, you're limited. Okay. If you produce intermediate copies, right, from the master copy, then you can simultaneously produce a lot of protein. You know. That could be one reason, right? But that's the way it is. Okay. Another reason could be, you know, and that's a theory, right? It's speculation that life may have originated in an RNA world where RNA would do all the work, all right? Because RNA can function, RNA can code information in the sequence of nucleotides, and it can also function as a catalyst by folding up, you know, in different shapes. So life may have originated in RNA world, and today what has happened is that, you know, but RNA is not a very stable molecule. So DNA has emerged as a stable carrier of genetic information, okay? Like if a guy commits a crime, right, then they burn down everything and all that, you know, still they might have left some trace of DNA, right? And you can go years later and dig that up, you know, and identify the suspect, right? RNA, you cannot do that. RNA, life may have originated in an RNA world, but later on, DNA emerged as the molecule for storing information, right? It's very good at storing information, whereas proteins emerged as the molecules for catalyzing reactions, all right? So in the beginning, RNA could do both, okay? Although it's not best suited for that because it's an unstable molecule. RNA you will disintegrate. Like if you go and, you know, with RNA, DNA doesn't matter. Even if the criminal tries to get rid of his or her DNA, it's not easy, right? They'll still get nailed down. With RNA, you don't have to do anything. If you go and touch it, right, it's gone because our skin and all, it has enzymes that degrade RNA, okay, RNases, okay? Because if you have RNA-coated viruses or bacteria that attack you, okay, you want to kill them, right? I mean, so if you touch something, you know, even with your bare hands, the RNA is gone, okay? It's all cut up, right? Well, DNA is different. So this gets back to your question. The use of an RNA intermediate makes it possible to more rapidly produce large amounts of a particular protein, right? Because you have a master copy, you copy into an intermediate copy, and then you do. It's like parallel processing, right? I mean, to facilitate parallel processing, you take the big code and you break it up into smaller units that you can run. So this is because many identical RNA copies can be made from the same gene. Each RNA copy can be used to simultaneously produce many identical protein molecules. Now, by controlling the efficiency of transcription and translation of the different genes, a cell could produce small amounts of some proteins and large amounts of others, right? Depending on the needs of the moment, if the cell needs a lot more of a particular protein, it will transcribe that particular gene and translate it, okay? If it doesn't need anything, it's not going to do that at all, okay? The code is there, but it's not active. In addition, a cell can change gene expression in response to the temporally changing needs for a particular protein. So over time, the needs for a protein might change, okay? And the cell can adjust its response, okay? It can produce more at the appropriate time.
Does it change the DNA?
No, it will not change the DNA. It will change the transcription, okay? The copying of the DNA into RNA. The DNA is the same, okay? Each and every cell in your body has exactly the same DNA, right? But the cells in your pancreas, the beta cells, they are the only ones that produce insulin. The red blood cells, they are the only cells that produce hemoglobin and so on, okay? So every cell is identical, but it's just that the genes that are active in different cells are different, okay? From the same organism, every cell is identical, right? And there are experiments that have been done to prove that. Now, RNA differs chemically from DNA. I mean, there are similarities, but RNA differs chemically from DNA in two aspects. The first one, and this is stuff that we already covered. The nucleotides in RNA are what are called ribonucleotides because RNA is ribonucleic acid. And so RNA, the nucleotides, they contain the sugar ribose, right? The 5-carbon sugar ribose. We talked about that, right? Instead of the sugar deoxyribose, which is present in DNA. So the sugars are different. And then the second thing is that DNA contains the 4 bases adenine, guanine, cytosine, and thymine, A, G, C, and T, while RNA contains the bases adenine, guanine, cytosine, and uracil. Okay. Now, since uracil or U pairs with A or adenine, the complementary base pairing properties described in the last chapter for DNA apply also to RNA, right? So therefore, in the case of RNA, the base pairing is adenine pairs with uracil, right, as compared to adenine pairing with thymine in DNA. And for the GC pairing is the same, right? Guanine pairs with cytosine, that's similar to DNA, right, even in the case of RNA. Any questions? Now, DNA and RNA differ quite dramatically from each other in their overall structure. DNA always occurs in nature as a double-stranded helix, while RNA is single-stranded. Unlike DNA, a single-stranded RNA chain can fold up into a variety of shapes, just as a polypeptide or a protein chain folds up to form the final shape of a protein, right? So therefore, RNA could also function as a catalyst, right? And it does in some reactions. So RNA can not only store information in the sequence of its nucleotides, like DNA, but it can also serve as a catalyst in certain situations or as a part of a larger molecular structure. The following figure on the next slide shows a single-stranded RNA molecule assuming a particular shape resulting from the formation of hydrogen bonds between complementary base pairs. So let me show you the picture. Yeah, this is the picture. So you can see that this is an RNA molecule. A is pairing with U through hydrogen bonds, G is pairing with C, and this kind of pairing all over the place is giving you a 3-dimensional conformation. Okay. Alright, so let's move on to transcription. So transcription means basically copying a part of the DNA into RNA, right? So what happens? During transcription, the cell is going to copy the required part of the DNA sequence into a nucleotide sequence of RNA or ribonucleic acid. To initiate transcription, first of all, you have the DNA double helix, right? So you have to be able to access the template. So the 2 strands have to come apart, right? So to initiate transcription, a small portion of the DNA double helix is unwound, right? One of the 2 strands, and so it's going to get unwound and the nucleotides over there are going to be exposed on the DNA. One of the 2 strands of the DNA double helix will act as a template for RNA synthesis, right? Now the RNA synthesis is also carried out by an enzyme, just like DNA polymerase. For RNA synthesis, we have an enzyme that is called RNA polymerase, which catalyzes the formation of an RNA strand that is complementary to the DNA template, right? And the process is similar to DNA replication by DNA polymerase and uses energy-rich nucleoside triphosphates, which are hydrolyzed to drive the formation of the RNA strand, right? Yeah, go ahead.
We have a 2 strand of DNA, but how to decide which one to generate?
I think either of them will work, correct? Yeah, that I don't know how exactly that is decided, you know. See, again, the other thing is that, you know, that complementary information, it has to produce protein, right? I mean, so it cannot just go and, you know, I think there'll be a promoter region, there'll be something that will signal, okay, where to start, okay. Now, however, transcription and DNA replication have some major differences. Maybe why don't you look it up, you know, like how do you decide which strand is going to be the template and which one is going to be the other one, you know. Look that up, you know, and come and tell us next time, okay. Look it up. I mean, I have not seen the answer to that question anyway, but, you know, my guess is that it would be the one that or it is possible that if you do because they are complementary information, okay? It is possible even if you do from the other one, it might be okay, so, but I'm not absolutely sure. Right. Now, transcription and DNA replication have got some major differences, right? First of all, since RNA polymerase synthesizes RNA, it will catalyze the linkage of ribonucleotides and not deoxyribonucleotides, okay, because it's synthesizing RNA, not DNA. Second thing is that unlike DNA polymerase, RNA polymerases do not possess nucleolytic proofreading activity. Last time I told you that if you have DNA polymerase, right, it always goes and checks its work, right, does some proofreading. If the previous one is not correctly base paired, it will cut it out. So there is nucleolytic proofreading activity. RNA polymerase does not have that, okay, and it's not necessary because RNA is temporary information, right? You need the protein, you made something from the master copy, right? But the master copy has to be preserved very well. So that's why DNA replication is so much more accurate, right? DNA polymerase has to do a much better job. Now, a consequence of this, that RNA polymerases do not possess nucleolytic proofreading activity, that means RNA polymerases could start an RNA chain without the need for a primer, right? Because it doesn't go and try to see what is the previous pairing, right? So it can start something from scratch. Also, transcription is a lot less accurate than DNA replication. In transcription, about one mistake is made for every 10,000 nucleotides copied, as compared to one mistake in every 10 million nucleotide copies in the case of DNA replication. Right? And this makes sense because the RNA thing is a one-time thing. Right? Even if you made mistakes and all that, it's not a big deal. Right? But if in the DNA replication there is a mistake, that is going to propagate, throughout the life of the organism and probably into the next generation too. So since the DNA serves as a permanent storage of information while the RNA is only a temporary intermediate, it is only imperative that DNA replication be much more accurate than transcription. All right, so now we get into some more details about transcription. So in transcription, the newly formed RNA strand does not remain hydrogen bonded to the DNA template strand. If you remember in DNA replication, what happened is that the two strands came apart, right? One was used as the template, then a new DNA strand was formed, and DNA replication is semi-conservative, right? You have one old strand and then the new strand, okay? Again, forming a double helix. In transcription, you don't have that. The newly formed RNA strand doesn't stay hydrogen bonded to the DNA template strand. Instead, the DNA helix will reform and it will throw the RNA chain out, okay? And as a result, the RNA molecules that are produced by transcription are usually single-stranded. Okay, it's a single-stranded molecule because there is no double helix here. Also, RNA molecules tend to be much shorter than DNA molecules because they are copied from only a limited region of DNA, right? You are just trying to get a copy of the portion that codes for a particular protein, right? As opposed to going and coding for the entire genome, right? So the RNA strand is going to be a lot shorter than, let's say, the entire genome, right? Also, the immediate release of the RNA strand from the DNA as it is synthesized means that many RNA copies can be made from the same gene in a relatively short time. Okay, so I mean, as soon as the RNA is released, okay, the portion, the gene that you're trying to transcribe, okay, is again available to RNA polymerase. Okay, RNA has been released. Again, RNA polymerase can get over there and make more copies. Okay, and it's moving along. The RNA polymerase is moving along the DNA as the transcription is taking place. So once it moves along, you could have another RNA polymerase get started. Right? I mean, it finished. Let's say processing this portion and moved on to the next portion of DNA. Then another RNA polymerase can come and start transcription here. Now, if you look at the RNAs that are typically present in a cell, all of them are produced by transcription, right? But not all RNAs are meant to be translated into protein. Because see, RNA, I said it has a function for storage of information, but it also can function as a catalyst, okay? So if there is an RNA molecule in there that is supposed to function as a catalyst, it's not meant to be translated into protein, right? It is going to work as a catalyst. So in fact, inside a cell, there are three types of RNAs that you can find. One of them is called messenger RNAs or mRNAs, little m RNAs. These are the RNAs that direct the synthesis of proteins. Okay, these are the RNAs that are meant to be translated into protein, right, in the next step, right? Then you also have what are called ribosomal RNAs, right, or rRNAs, little r RNAs. They form the core of the ribosomes on which mRNAs are translated into proteins. All right, the machinery inside the cell that is responsible for translating RNA into protein is called a ribosome. And towards the end of this chapter, we will come across the ribosome, see what it looks like and things like that, how it functions. All right, but for now, just keep in mind that when ribosomes are made up of some proteins and also from some RNAs. Okay, and the RNAs that are present in ribosomes, they are called ribosomal RNAs. These are not translated into protein. Okay, they maybe are folded up to give the ribosome the particular shape, right? And then the third category of RNAs, these are called tRNAs or transfer RNAs. They form the adapters that select amino acids and hold them in place on a ribosome for their incorporation into proteins, right? These are like the decoders, okay? Like, because remember, this is the whatever, 4-letter language of DNA. Okay, from DNA you're copying into RNA. It's still a 4-letter language, right? But now that information in that RNA, right, the letters that you see, that has to map into the 20 amino acids. Okay, so you need an adapter molecule or a decoder, right? Like Viterbi decoder or whatever, for communication people. So, there is a decoder that is needed because this information on the RNA strand, right, RNA that has been synthesized, there has to be some decoder that will go along this, look at, the different codes, right, and stitch together the right amino acids. We will discuss that in detail in this chapter. Okay, I'm getting to that, right? But the RNAs that are responsible for that, they are called tRNAs or transfer RNAs. Now, in the case of eukaryotes, what are eukaryotes? Anybody remember? Cells with what? Yes, good. Nucleus and other organelles. So in eukaryotes, each gene coding for a single protein is typically transcribed into a separate mRNA molecule. Right? For each gene, you are going to have a separate mRNA molecule. In the case of bacteria, a single mRNA is often transcribed from several adjacent genes and therefore contains information for several different proteins. Right? So you might have one mRNA molecule that has got information for many different proteins. Right? In humans and in other eukaryotes, it's usually just one gene, one protein, right, and one mRNA. Now, the way in which transcription is initiated differs somewhat between prokaryotes and eukaryotes, right? In the case of eukaryotes, it's a lot more complicated. Okay, it's like a committee. Okay, a lot of things have to kick in, right? And the committee gives you a positive vote, then you move forward. Okay, that kind of thing. In the case of prokaryotes or bacteria, it's a lot simpler. So in this chapter, I'm going to talk only about prokaryotes. Next chapter, we'll look at eukaryotic transcription. Right? Now, in prokaryotes or in bacteria, the RNA polymerase molecules will slide rapidly along the DNA. And the molecule will tightly latch onto the DNA when it encounters a special region that is called a promoter region, which contains a sequence of nucleotides indicating the starting point for RNA synthesis. So it's going over there, suddenly it sees the promoter region, it knows that that's where it's going to latch onto the DNA and try to get started. Right? So then, once it hits the promoter region, it opens up the double helix immediately in front of it to expose the nucleotides on a short stretch of DNA on each strand. Right? Open the double helix, and one of the two exposed DNA strands then will act as a template for synthesis of the complementary RNA. Okay. Now the RNA chain elongation will continue. The chain is being synthesized, it keeps on growing until this RNA polymerase encounters a second signal in the DNA. It's like a stop signal, right? The terminator or stop site. There is special code for that. When it starts also there is a special code, okay? It's like communication codes, like start and stop. There are codes for that, okay? And all that has been mapped out. It was mapped out in the 1960s, all right? People have mapped out which code corresponds to starting, which one to stopping. And the people who did those, each of them, they got Nobel Prizes, okay? This is a big discovery, So the RNA chain elongation continues until the RNA polymerase encounters the second signal in the DNA, which is the terminator or stop site. Here the polymerase will halt and it will release both the DNA template and the newly made single-stranded RNA chain. Now I give you the picture here. I think it will be better to talk with the picture, to describe with the picture. So this is an example of a double-stranded DNA, see 5' to 3' running from left to right on the top strand, then this is 3' to 5'. This object here, this blue object, that is RNA polymerase, okay? So RNA polymerase is sliding along the DNA, right, in the case of a bacteria, and then it encounters this promoter region. Suddenly it sees the promoter region, it latches onto the DNA. There is a subunit of bacterial polymerase, that yellow ball over there, it's called the sigma factor. It is thought to basically help in the identification of the promoter region. Okay. So this goes over here, and then after, so it detects the start site, it starts producing the messenger, producing the RNA, this blue stuff, all right, in here. It's producing the RNA. After about 10 nucleotides have been synthesized, then this sigma factor drops off, right? It's gone, and this thing continues. So sigma factor is something like, it's like starting a car. You just need the battery to start the car, that kind of thing. Okay, it's just for the starting. Once the thing starts transcribing, then the sigma factor goes off, and then the RNA polymerase continues. And you can see that it is synthesizing the strand. Okay, as this one moves, the strand continues to grow. All right, until it finally reaches the stop site. And here the bottom DNA strand is the template. Okay, see that one is running 3' to 5' here. This one is running 5' to 3' here. It's complementary. Okay, so it keeps on going this way until it goes to that stop site. Okay, so there it'll, as soon as it hits the stop site, it'll release the whatever has been synthesized so far. Right, it'll release that RNA. And it'll also let go of the DNA, and the DNA double helix forms, and then it is now free to be bound again by another sigma factor, right? And then again go looking for a transcription site. Okay. So this is explanation using the figure, and I'll just go over the okay. So once the RNA polymerase reaches the terminator or stop site, the polymerase is going to halt and release both the DNA template and the newly made single-stranded RNA chain. Then, as I mentioned, using the figure, a subunit of bacterial polymerase, which is called the sigma factor, that yellow ball in the figure, is primarily responsible for recognizing the promoter sequence on DNA. Once the DNA polymerase has latched onto the promoter and has synthesized approximately 10 nucleotides of RNA, the sigma factor is released. The RNA polymerase then moves forward and continues the transcription process. And after the polymerase reaches the terminator sequence and is released, it reassociates with the free sigma factor and is ready to start transcription again from a promoter region. Okay. And the figure that I showed you is a schematic diagram showing an RNA polymerase molecule transcribing a bacterial gene. And here, as I emphasized, the lower DNA strand serves as the template for RNA synthesis. Okay. And you're still supposed to find out, how they decide which strand it is going to be. Okay, the lower one or the upper one. Here it's the lower one. Okay, I can tell you because looking at this, it goes 5' to 3' like this, so that one has to go 5' to 3' this way. Yeah, go ahead.
Sigma factor corresponding to different protein.
No. Sigma factor is just, it's like something, missing from the RNA polymerase, okay? To complete it, all right? That's the sigma factor, right? To just get it started, right? Yeah, it's not different for different proteins, right? It's the same. Now, transcription can only proceed in the 5' to 3' direction for the synthesized strand, and that is clear from this figure, right? See, it's 5' to 3'. It's going in the 5', it'll start, go in the 3'. So transcription can only proceed in this direction, right? Okay. And again, if it was the other strand that was being used, then it would have to go in this direction, right? Maybe that has something to do about which strand is going to be used, okay, as a template. Moreover, a portion of DNA can be transcribed only if it is preceded by a promoter sequence, right? Because the RNA polymerase is going to go and look for the promoter. If there is no promoter, no transcription will take place, right? So this ensures that unnecessary transcription does not take place. Okay, the cell should not be transcribing anything just for the fun of it. Right? It should be transcribing only when something is needed. So if there is a promoter region, that's indicating that that gene should be transcribed, right, or could be transcribed. That's when transcription would take place. Right? If there is no promoter region, it will not take place. Right? Now, since prokaryotes or bacteria, they don't have a nucleus, prokaryotic transcription takes place in the cytoplasm. Right? There is no nucleus, so everything is the cytoplasm, no nucleus and organelles. And as mRNA molecules are transcribed, ribosomes or the protein making factories, they immediately get onto the 5' end of the RNA transcript and protein synthesis starts. Okay, so if you go back to the previous picture, as this thing is being transcribed, right, now it needs to be translated. So the ribosome will get on the 5' end and start translating from here, again along this direction. Okay. It'll do the decoding kind of, it'll take these letters 3 at a time and then map them to amino acids. Okay, and we'll discuss that in detail very shortly, if not today, next time. Now in eukaryotes, on the other hand, the DNA is enclosed within the nucleus, right? I mean, in eukaryotic cells, you have a nucleus and other organelles. Most of the DNA is inside the nucleus, right? Except some in the mitochondria, but most of it is inside the nucleus. So the transcription, okay, which is directly copying from the DNA, that will take place inside the nucleus, right? However, protein synthesis takes place in the cytoplasm, right? So that means the RNA that is transcribed inside the nucleus, in the case of eukaryotic cells, it has to be moved out of the nucleus and into the cytoplasm, okay, before translation can take place. Now, before RNA is exported from the nucleus, it goes through several different RNA processing steps, right? And depending on which type of RNA is being produced, these transcripts are processed in different ways before leaving the nucleus. In the case of eukaryotes, the RNA that is produced by transcription but hasn't been processed as yet, right, before being exported, is often called the primary transcript. So this primary transcript needs to be processed before it's sent out, out to the cytoplasm. Now the primary transcripts that are destined to become mRNA molecules. Again, mRNA molecules are those molecules that are going to get translated into protein, right? So you have mRNA, you have ribosomal RNA, and then you have also tRNA. So primary transcripts that are destined to become mRNA molecules, they undergo the following two processing steps before being exported from the nucleus. The first one is the addition of a methyl guanine or a methyl GCAP. So I've shown you the molecule guanine, all right? At some location you put a methyl group or CH3, right? Group, and you get methyl guanine, okay? So methyl guanine is added at the 5' end, right, of this RNA, which usually occurs just after the RNA polymerase has synthesized the 5' end of the primary transcript and before it has completed the transcribing the entire gene. That's one modification. So at one end you're going to have a methyl guanine cap at the 5' end. At the 3' end of eukaryotic RNAs, the 3' end is trimmed and there is a large number of A's or adenines that are added over there. And then a series of repeated adenine A nucleotides are added at the cut end. And this processing step is referred to as polyadenylation, right? So at one end you're going to have a 5' methyl G cap, at the other end you're going to have a polyatal. This is like, parity checking in communications, okay, where you send some parity bits, okay? Because when the message is received outside, okay, is it intact or not? You can look at the two ends. If it's missing the methyl G cap or the polyatal, means something might have gone messed up, right?
You mean extra molecule is added to the RNA?
Yeah, on the tail, yes, yeah. These are extra molecules that are added, yeah. Yeah. And only for messenger RNAs this thing is done, okay? So if you want to distinguish between messenger RNAs, ribosomal RNAs, and tRNAs in a cell, you can look for the polyatal because the messenger RNAs will have the polyatal, okay? Or the 5' methyl guanine cap. So the trimming and nucleotide addition steps are carried out by two different enzymes. The sequence of adenine nucleotides is referred to as a polyatal and is generally a few hundred nucleotides long. The two modifications, capping and polyadenylation, are thought to increase the stability of the mRNA molecule and to aid its export from the nucleus to the cytoplasm. Right? And they also later serve to indicate to the protein synthesis machinery, that is the ribosome, that both ends of the message are present and therefore the message is complete. The message has not been corrupted. Yeah, go ahead. It can vary, I don't think it'll have exactly the same length, but maybe a couple of hundred nucleotides, a few hundred nucleotides long, yeah. It may not always have the same length, so I've not seen that anywhere there's a concrete length, but a few hundred nucleotides. But that'll be the characteristic, okay. So these are like the two ends that of the message. If the two ends of the message are intact, okay, you're reasonably confident that.
If it does not have like a fixed number of nucleotides, how do you know that, I don't know, that it's intact? I mean, like in one side you have just one methyl
GCAP, yeah.
On the other
But
side.
yeah, I don't know where the cell makes the cutoff to decide that it's not intact. But cell has mechanisms. If the RNA is not produced properly, it'll chew it up, and even if the protein is not produced properly, right? So it has some, some kind of check. Okay, I don't know the details of how that happens, and I'm not even sure that people have figured those out. Now, unlike bacterial genes or prokaryotic genes, eukaryotic genes contain both coding and non-coding regions. Remember, I told you out of the 3 billion nucleotides, only about 2% is coding, right? The coding regions, they are called exons, and they are interrupted by non-coding regions, which are called introns. Right? The exons are usually much shorter than the introns, and the coding portion of a gene is often only a small fraction of the total length of the gene. Right? Even for the human genome, out of the 3 billion letters, only about 2% is coding for anything. Right? The rest is non-coding. Now, for RNAs that are destined to become messenger RNAs, right? You want a continuous message, right? So that stuff is going to be cut out, right? The non-coding portion will be cut out. So the introns are removed after capping and polyadenylation, and this is carried out in the nucleus before the messenger RNA is sent out to the cytoplasm. And this process is known as RNA splicing, right? This is another important terminology and will result in a functional mRNA molecule with uninterrupted coding sequences. And the RNA splicing also, another big thing is that the splicing will not take place the same way all the time, okay? Because you could have a number of exons with introns in between, okay? The question is, when you cut out the introns, are you just going to cut out introns, or are you also going to cut out some of the exons in between, okay? So you could get some different molecule too. And I'll show you, it's there on the next slide or the one after that, how that's going to happen. And they actually refer to it as alternative splicing. Actually, not next slide, but in a couple of slides I'll get to it. Now, a natural question comes up. How does the cell determine which parts of the primary transcript should be removed, right? Which are the non-coding regions that should be removed? Now, as it turns out, each intron contains a few short nucleotide sequences which act as cues, right, or hints for its removal, right? And these sequences are found near each end of the intron and are the same or very similar in all introns. So introns are removed from RNA, again by some other proteins or enzymes that are composed of a complex of protein and RNA. And these splicing enzymes are called SNRPs, SNRNPs, or small nuclear protein particles. Right. So these guys go in there and they basically cut out the non-coding region and put make the coding region contiguous. Now the presence of yeah, go ahead.
Is there a function for the intron?
Well, I mean, see, it's not known, right? Like it may have had functions many thousands of years ago. We haven't figured out the function, okay? But parts of it, there could be some, location where, there might be some micro RNA might bind or something, because it's really quite complicated, okay? I mean, it's not just the DNA, okay? There is a lot more information that is needed. Like you could have the information in the DNA, but then some parts of it could be methylated. It would change the activity of the DNA, right? So it's a lot more complicated than just engineering systems, But, the non-coding region, people used to call it junk DNA, okay, because it has got no function. But they're finding out that it does have function. It's just that we haven't figured it out, right? So people have a long way to go to figure out the entire mystery, right? Now, the presence of numerous introns in eukaryotic DNA makes genetic recombination between exons of different genes more likely. See, because here you have exons and you have introns, okay? The intron is not a functional part, okay? It's like non-coding region. So if you want to have genes crossing over and things like that, if everything was functional, you have crossover, you're going to lose function, right? But if you have regions that are not that crucial, that makes it possible to combine DNA in many different ways, without disrupting the primary function, right? And we'll see examples of that later because there is a chapter on genetic variation and recombinant DNA technology that will come up later. So I will talk about these things in more detail later on, okay? But for the time being, just note that this increases the possibility that even when you have genetic recombination, you're going to have functional genes producing functional proteins, right? So here we note that eukaryotic genes for new proteins could have evolved quite rapidly by the combination of parts of pre-existing eukaryotic genes. Moreover, the primary transcripts of many eukaryotic genes containing 3 or more exons can be spliced in various ways to produce different messenger RNAs depending on the cell type in which the gene is being expressed. This is referred to as alternative splicing, and I think that is best illustrated, not this one, I think it's probably, okay. Um, No, this, yeah, first let me just explain this picture and then we'll go to the, I'll talk about alternative splicing. So here you have an exon A, all right, that's coding, there's a non-coding region, and then there is an exon B, right? So the SNRP, or that small nuclear ribonuclear, that protein particle, it'll go in there, all right, inside the nucleus, and it'll basically, cut this fellow out, this thing in the middle, it'll cut the intron out and stitch the two of them together to make one continuous RNA molecule, right? A continuous coding RNA molecule with exon A and exon B, right? So at each intron, a group of SNRPs assembles on the RNA, cuts out the intron, and rejoins the RNA chain, releasing the excised intron as what they call a lariat. Okay, this thing will be released to the inside the nucleus. Now here, I don't have that in this picture, but let's say you had exon A, then there's an intron 1, exon B, intron 2, right? And then there's an exon C. So let's say there are 3 exons, all right? So you have 2 possibilities, okay? You could cut out the 2 introns here and you could get one protein that has exon A, B, and C, all right? Another possibility might be you just cut out this intron along with this exon and that intron, and you have only exon A and exon C, all right? So they will be different proteins with different properties, okay? So the splicing can be done in alternative ways. That's a big topic, okay? Alternative splicing, because and the cell uses that mechanism, right? I mean, that from the same gene, right? It can produce different proteins depending on the needs, depending on the context, depending on the cell type, what type of cell it is. Now the length of time that an mRNA molecule persists in the cell affects the amount of protein that is produced from it, because the mRNA molecule has to be around in order for the information on it to get translated into protein. So different mRNA molecules have got different lifetimes, right? So some of them might have a short lifetime, some of them might have a long lifetime, and different amounts of protein are produced. So although the genomes of current day prokaryotes do not contain any introns, it is thought that their earlier ancestors may have had introns in their genes, right? And the prokaryotic cells, the bacterial cells, may have evolved and gotten rid of their introns in order to have a smaller genome, which would allow faster DNA replication, thus facilitating very rapid reproduction. Because like bacteria like E. coli reproduce every 20 minutes, okay? You don't want a large genome, so you're better off getting rid of all the junk DNA or non-coding DNA, and they have gotten rid of that. That's what they think, nobody knows for sure. All right, let's stop here for today, right? I mean, we're done with transcription. Next time I'm going to do translation, right?